Assalamualaikum dear students. I'm Dr. Uzma and today we are going to discuss the different forms of the DNA. Basically we really knew that there are three forms of the DNA, but I would like to tell you few more forms of the DNA so that whenever a person in future is going to ask you what are the different forms of the DNA, you could able to elaborate and give them a great depth of the concept. Okay, without wasting time, let's get started. These are the different forms of the DNA. As you can see, that is the A form, B form and the Z form. But these are not only the form that exist in a cell, or there are some other form like a cDNA, ddNA and eDNA. DNA possesses 6 different forms that are either in a left-handed or a right-handed fashion under appropriate physiological condition. There are some unusual structures like the bent, triplet strand, four-stranded DNA. If you don't understand what is the meaning of bent, triplet and four-strand DNA, please kindly watch my previous lecture. Over there I have explained you each and every term. Okay? Just now I'm just giving you what are the terminology of what are the unusual structures that occurs in the DNA. Okay. The conformation of the DNA strongly depend on what is the pH and what is the ionic strength of that solution. And we can see the bacteriophage Cy174 it usually have a single-stranded DNA, and the viruses and all prokaryotes have a circular and double helical DNA. Before starting, what are the dimensional form of the DNA, three structures and all? Let's take a look what is the DNA and what is really a DNA. It is mainly a flexible molecule and it has a number of bonds which have a rotation around it like a sugar phosphate backbone, and it has a thermal fluctuation which produce bending, stretching and unpairing. Unpairing melting means at high temperature how the structure of the DNA melt. I will let you know its diagram along with a graph in the future lecture. Till now you just know what is the unpairing and the melting is. Just this terminology. Many significant deviations of the Watson and Crick DNA structures are found in the cellular DNA, and some and all plays an important role in the DNA metabolism. There is a structure variation and which defined by the Watson and Crick base pairing, which shows the complementary strand complementarity and the anti-parallel strand. Anti-parallel strand means one should be a purine, the other should be of pyrimidine, not like the purine and purine, right? And it have a complementary strand complementarity, that is the AT and the GC. It has a two double bond like of hydrogen, and GC has a triplet bond, and triple bond of hydrogen. What is the structure variation that reflect the three things? What is the different type of possible conformation of deoxyribose? What is the rotation that cause the contiguous bond that make up the phosphodeoxyribose backbone? And what is the free rotation at the C1N glycosyl bond? All these terms I have already explained you. Okay. Because of steric constraint. Steric constraint means there is some sort of steric hindrance. Like in the purine nucleotide are restricted to two stable conformations with respect to the deoxy, that is the zinc and the anti. And the pyrimidine has a restriction of anti-conformation because of steric hindrance or steric interference between the sugar and the carbonyl oxygen at C2 position of the pyrimidine. The Watson and Crick structures. It shows the B form DNA. The B form is the most stable because of its random sequence in the DNA molecule, and because of this feature it is a standard point of reference in any study of the properties of the DNA under physiological condition. Let's take a look why the DNA has different type of conformation and why DNA adopt all these things. because of the DNA sequence, chemical modification of bases, the type and the concentration of metal ions in the solution, salt concentration, and the quality and the direction of the supercoiling. These are the different forms of the DNA, that is the A, B, Z, C DNA and D DNA. This is the perpendicular view of the helical axis of the DNA conformation, and this is the view of the helical axis. You can easily understand now. The A form. A form give rise to a ribbon-like structures with an open cylindrical core. I hope you know what is the meaning of the core slender, right? And it is mainly present in a solution that is relatively devoid of water. And the DNA is arranged in a right-handed double helix structure, and its helix is wider with an 11 base pair per helical turn, and it has a base pair tilt about 20 degrees with respect to the helical axis. Form also both the strands are anti-parallel or they have a plectonymic coiling. This is the plectonymic coiling. This is the DNA is also a metastable, that is, it can easily turn into the D form. The major groove is narrow and more profound, whereas the minor groove is wider and flattened. The sugar is puckered at C3 endoconformation. And also note down a point that the DNA is a more stable because of an additional of an hydroxyl group. This is the B form structure. I have told you what is the major and the minor. So this is the major groove and these are the minor groove. For better graphic, kindly increase the resolution. Okay, this is the B form. It was pioneered by Watson and Crick, and is the most common form of the DNA, and it is also a right-handed double helical structure, and under normal physiological condition it is 92% relative humidity and ionic strength. And when we do the extra diffraction, we can able to understand what is the average conformation of the DNA. And the majority of the DNA comprises the bDNA form, and its coiling is a right-handed pattern because it has a right-handed double helical structure. The nucleotide bases occupy the core, where the sugar phosphate backbone occupies the peripheral region of the DNA helix. In solvent, at the edge the bases are present, and it has a diameter of the helix is 20 angstrom, and its single turn is 10 base pair, which is perpendicular to the helical axis. There is also a plectonymic coiling in the bDNA, where the two strands intervened at the same helix. The B form structure is asymmetric as the major and the minor grooves are present. So the major and the minor grooves are wider and narrower. And it has a sugar pucker at C2 endoconformation. Dear student, I am just letting you the terminology. What is a C2 endo and C3 endo exo concept? Please learn, or if you don't know, watch my previous video. The width and the depth of the B-type major groove is 12 angstrom and 8.5 angstrom, while the width and the depth of the B-type minor groove is 6 angstrom and 7.5 angstrom. Now let's take a look on the Z form. It was first introduced by the three scientists, and their name is Rich, Nordim and Yang, in 1984, and its structure is a left-handed double helix. There are 12 base pair per helical turn. Therefore, its structure appears to be a slender, elongated or a zigzag appearance. By the name zigzag, you can get the name Z form. This form is mainly found in alternating purine and pyrimidine sequence. Nowadays, it is stabilized by the high salt concentration. It shows asymmetric structure configuration which give rise to the zigzag helical structures with no internal space. Its helical diameter is 18 angstrom and its base pair tilt is 7 angstrom. In Z form also there is a zigzag plectonymic coiling. It possesses an anti-parallel structure like the bDNA, and it has a sugar puckered at 3 endoform of the purine base and C2 endoform of the pyrimidine. The Z form is mainly found in a place where the concentration of salt, or we can say the concentration of cation and anion, is more. There are some other form of the DNA along with the B and Z, A, B and Z. There is the C form, D form and E form. It is found in a minimal quantity, approximately 66% relative to the humidity along with the ionic strength, and it has a right-handed fashion helical turn with a helical pitch of 30.97 angstrom. It consists of 9.33 base pair per turn, and both the strands of the cDNA are antiparallel to each other unlike the zDNA, and its helix diameter is 19 angstrom. Rotation base pair it's 38.6 degrees and the base pair tilt is 7.8 degrees. The size and the shape of the cDNA are smaller than that of the bDNA and aDNA. D form in the cell, the D form is found very rarely, and the D form is twisted in a right-handed fashion, and it is known as the poly-DADT or poly-GC and DC form. The D form consists of 8 base pair per turn, which are displaced backwardly with respect to the DNA helix. Both the strands of the ddNA are antiparallel to each other, and the base pair tilt is negative 16.7 degree with an axial rise of 3.03 angstrom base pair. The other form is the E form. In the E form, the eDNA is found very rarely and having an extended DNA. The helix of the eDNA is long and the base pair are perpendicular to the helix axis. Dear student, this is the same point. Please, okay. eDNA consists of a D major eDNA consists of a deep major axis and shallow minor axis. The X-ray crystallographic, the eDNA is found as an intermediate between the bDNA and the aDNA. Okay. Now let's take a look what the factor influencing the conformation changes in the diagram. The major factor is the physiological condition, the second is the DNA base sequence, and the third is the presence of the protein. So the conformation changes of the double helical DNA structures, there are the three factors. Physiological condition, the humidity and the ionic and the hydration environment favors the conformation change in the DNA. The second one is the DNA base sequence. It is the most crucial factor which decides the size, shape and coiling and other structural properties of the DNA. The presence and the binding of the protein. A protein binds with one helical conformation of the DNA and it changes its structure into the different form. For example, a protein can bind with the bDNA and change the conformation into A and Z form. This is a very simplified chart of the properties along with the different forms of the DNA A, B and Z. Some points are missing, so I have added one more chart so that you can better understand. So the first point is the helical sense. It is a right-handed coiling, right-handed, and the Z form is a left-handed coiling. What are the occurrence conditions? It is a 75% relative humidity because of the presence of sodium, potassium and cesium. It is a 92% relative humidity, but it has a low salt concentration, the B's DNA. In the zDNA, it occurs at a very high salt concentration with alternating purine and pyrimidine sequences. The plane of the base, they all have a perpendicular to the helical axis. The rotation base pair 33, 36 and 30 degree. The axial rise per base pair 2.56 angstrom, 3.38 angstrom, 3.71 angstrom. The next one is the helix diameter, which is 25.5 angstrom, 20 angstrom and 18 angstrom. The base pair per turn, it is 11 base pair 10 base pair and 12 base pair. The sugar phosphate backbone. It has a normal, and they have a zigzag. By the name zigzag, the Z-DNA gets its name. The base pair tilt is 19 degrees, 6.3 degrees and 7 degrees. Helix pitch, which is 25.5 angstrom, 35.5 angstrom and 45.6 angstrom. Major groove. They have a narrow and deep major groove. The B form have a wide and deep major groove, and the Z form has a flat major groove. Minor groove. The A DNA has a wide and deep minor groove. B DNA has a narrow and deep minor groove, and the Z DNA has a narrow and deep minor groove. Sugar puckering. C3 endoconformation. B DNA has a C2 endoconformation, but the Z DNA has a C3 endoconformation for purine and C2 endoconformation of the pyrimidine. One point is missing, that it also has a glycosyl bond conformation. The A form has an anti-form, the B form has an anti, but the Z DNA has an anti for the pyrimidine and sig for the purine. Points to remember: that A DNA, its position is uncertain, so there are evidence of the short stretch of Z DNA in bacteria and eukaryote. A DNA is reported is in vitro, while the Z DNA is reported in vivo, like in bacteria and eukaryote. Helical pitch means per helical turn length. Okay, I hope this video gonna help you. Thanks for watching my video. Stay tuned. We will be continued soon. Thank you for watching.